kind of black and white picture, but I fear that I can give you no black and white picture. There is, in the end, 50 shades of gray, or whatever you call that. The main problem is that we deal with a very complex virus where we still lack knowledge. This virus is, in the end, out of Africa. It is, in the end, optimized for blood transmission. So whenever it comes to these oral routes that we usually encounter, it's in the end crippled. So it's rather inefficient, but it's very stable. So most of the transmission pathways you also ask for, they are not very likely. You need quite a lot of virus to have a good oral infection in a lot of circumstances, but the virus is very stable. And if you try 100 million times, it will work 3 times. And that's the main problem. We deal with residual risks, but these residual risks put us at stake. And in the end, any introduction is a catastrophe. And for foot and mouth disease, Dr. Rieder, it's a bit easier because we know the virus is also whatever transmitted airborne and so on. For ASF, it's not so easy. It's not highly contagious in a lot of circumstances, but it's still giving us a headache.
Wonderful, thank you. And then Dr. Rieder, if you'd like to elaborate a little bit more about some of the topics that came up in the Q&A.
Yes, thank you. I could just recall now one regarding the transmission of the leaderless. In our safety experiment. We did several inoculations and experiment to just look into the safety of the live virus when inoculated in susceptible animals. And we use the more commonly and more sensitive route of inoculation. And for instance, the heel bulb of swines are very sensitive. And when we did that immunization with different strains of the leaderless marker vaccine virus live, we didn't even detect the virus in the nearby lymph nodes. We didn't detect seroconversion on the animals, and we didn't see lesions. Going into the cattle experiment, when we did the inoculation in the tongue, which is highly sensitive, we didn't even see the mark of the needle, at the point of inoculation, indicating that it was so restricted in replication that we didn't see any replication locally. Therefore, we didn't observe viremia and shedding. That was the question, what we have observed for transmission in cattle and pigs, and our results are on experimental settings in the laboratory using these highly sensitive routes, and we put animals in contact, and in the case of cattle, we look also at the virus in the air, in the rooms of where the animals were hosted, and we didn't see any detection of the virus. So I think that was responded also on in the Q&A box.
Wonderful, thank you. Thanks so much for that follow-up. And then Dr. Wallace, if you noticed any particular themes in the questions that you received in the Q&A box, I'd love and really appreciate your elaboration on any of those if you'd like to go ahead.
Thank you, Hayley. If it was up to me, I would talk about the wildlife aspect all day long, but no, I think the questions were quite diverse. So the one that maybe could require a bit more elaboration is the very, very pertinent question about pre-existing immunity to the vector. So in other words, if animals either been vaccinated or pre-exposed to a natural infection with either like lumpy skin disease virus, sheeppox virus, or goatpox virus, won't that interfere when the time comes, hopefully not too distant future, when one or more of these recombinant vaccines are actually ready for use in the field. And so certainly a lot of evidence suggests that there can be some interference when this pre-existing immunity to the actual vector. But we're not too worried about this, because what we reckon is, of course, initially, when one of these is used in the field, yes, there could be a bit of interference, but as new animals are actually then inoculated for the first time, or first time exposure to the vector, again, with the recombinant proteins being expressed, then that won't be an issue. So as time goes on and the recombinant vaccine is used more and more, especially as a first time, then the issue of pre-existing immunity will become less and less of an issue, and then eventually, hopefully, almost not an issue at all. But of course, again, wherever there's natural outbreaks and things, that would always be potential concern. But that's just something one has to deal with. And so I hopefully made that a little bit clearer, if possible. Thank you.
Yeah, fantastic. Thank you so much, Dr. Wallace. And I noticed that Dr. Geary is back online with us, so I will graciously hand the floor back to him. Thank you, Dr. Geary.
Yeah, isn't technology great? So I had everything on time and froze up. So thanks, Haley, for doing this, except I don't know exactly where you were with things.
Oh, yes, of course. So just to catch you back up, we just went around the floor briefly and allowed everyone the opportunity to provide a little more context for the things they were seeing in the question and answer box.
Okay, well, another little kink in the system is that when I had to restart everything, I lost all the Q&A. Is there any way you can send that to me?
Yeah, so I would also invite all of our attendees online to go ahead and submit some additional questions for our speakers. And I will follow up with you, Dr. Geary. I'll send you some additional ones. You really haven't missed too many from the beginning of the break.
Well, I had some that were on that list that I wanted to ask, but anyway.
Oh, well, how about this? I'll go ahead and ask the first one for you, Sandra, and then I'll send you, Dr. Geary, the text of the other questions for
That'd be good.
later.
That'd be good.
So the first question from the chat for you, Sandra, someone's thanking you for your fantastic and fascinating presentation and asks if you could please update the audience on the ASF crisis in China that is impacting the world economy and possible political stability. The headlines are not commonly brought to public attention in US news media. And then are RNA vaccines, those that are antigen coding or RNAi, an option for ASF? Why or why not?
Okay, so we have already covered part of the this Chinese situation, and at least what I can do in a politically correct way has been said, I guess. So regarding any kind of what you would call a DNA or messenger RNA vaccine, in the end, I'm not aware of approaches regarding the messenger RNAs itself. It is a bit also a problem that we do not really know the protective antigens that would give us a nice T-cell immunity. In silico predictions were not really successful when put into vectored vaccines, at least. The DNA vaccines have failed to provide for protection. I think there's still room for improvement, and this is something we should still tackle and look if we can find the right targets. But it is so far still difficult because of the lack of knowledge regarding the antigen that would give us more than antibodies that are not protective. Regarding the sorry.
Go ahead. Go ahead.
No, I just saw the other question, but I don't know if you want to pose it.
I'm still getting this technology under control. Go ahead. Okay,
there was the question from Luis Rodriguez about the in the end use or the envision of. I can read it aloud so that everybody has it. It's given the experience we are living with COVID vaccines deployment under emergency use only before full licensing. Do you envision a similar situation for ASF live attenuated vaccines, such as the I-170L live attenuated vaccine candidate from Plum Island? Regarding this question, we have to see that this ASFV candidate is now going for field trials and also possible licensing in Vietnam. I know that the first field trials have been conducted. It showed again that under field conditions, the situation is a bit more complex than in our experimental settings, but it is still a very promising candidate. And I guess if the first field trials give us a good result, I could envision the use of such a vaccine also under other circumstances. The question is, what is the, how would we decide that we need it? In Central Europe and probably also in the US, a single outbreak of African swine fever in domestic pigs would not trigger this kind of approach. And for the wild boar, where we would probably use it in Central Europe, there we at the moment do not have something that is field deployable in terms of a baited vaccines. We have bait candidates, but it has never been put to practice and not even in a proof of concept. So yes, I think that we could have a similar situation, but this would mean that we have really a grave situation for ASF coming up.
Okay, thank you. So one of the other questions that the audience had is, can you provide any thoughts on how long ASF can persist in soft ticks, and the challenges that might present for vaccination efforts?
It depends on what you're talking about. In the cycle in Africa, it's in the end something that that goes on and on, because they are competent vectors of, if you have soft ticks of the Ornithodoros genus. There you would give it on the larval stages, in the eggs and so on, so you would keep it. A lot of other ticks and also arthropods have shown to be non-competent. There is some kind of mechanical transmission thinkable, but in the end, this is then only mechanical. And there, at least from our experience, we talk about days, even in part of the worst case. It is for, if you have competent vectors of the Ornithodoros genus, then you really run into trouble. And this was one of the reasons why it was so difficult to control the disease in Spain and Portugal, especially in the Iberian pigs, where the soft ticks were involved.
Okay, thank you. We'll stay with you for a minute then, okay? Could you speculate on reasons behind why vaccines you've tested differ so much in efficacy and shedding?
Very good question. We still try to find out. In the end, the vaccines I tested, they had different deletions. And what we can say is that the deletions in the multigene families 360 were really highly efficacious. Other regions that are also implicated for violence, 9GL and these kind of UK genes, they also gave protection, but there we saw more residual violence and also more shedding of both the vaccine virus and later on the challenge. At the moment, it's speculation, and ASF is also there giving us a headache. If you do successive deletions, the combination of two attenuating deletions does not always result in more attenuation. And if you put the same deletion to another virus strain, it does not result in the same phenotype. So it comes to this, to what I said, we are still lacking knowledge. But what we know is that at the moment, all the promising candidates have either multigene family deletions or some deletions that we we just did, or we don't really have the full background. The promising candidate from the USDA at the moment, the I-107L, if you ask Manuel Walker, what is this exactly what you deleted? This is not, so, he will not be able to completely tell you the story. It is in ASF, a lot of things are still a bit try and error, and yeah, looking in analogy from other viruses.
Would Manuel not be able to tell you? Are you saying that he isn't certain, or is this because this is patentable material?
There's also some patentable material, but in the end, we do not completely know what it does.
Okay. Well, thank you. So I want to continue with the questions from the audience. And after we've exhausted most, if not all of those, then I want to turn to a discussion of what the future may hold for animal vaccinology and just more prognostications. And I'm hoping that the audience will also weigh in on these things. If you have thoughts on what you think the future may hold, write those in also, and we'll bring them up for discussion. But for the time being, I think Sandra will ask you one final one before we move on to someone else. And what's your overall feeling as to whether we'll be able to develop a safe and efficacious vaccine against the Georgian strain in the next 5 to 10 years? So you get to predict on that.
Very nice question, Chris. But yeah, if you would have asked me 2 years ago, I would have said it's very difficult, and I don't have a good feeling at all. I have now I did this comparative trials with the different candidates, and there were at least 2 of them that I would call a vaccine. They provided full clinical protection, almost sterile immunity. And what I have to look into it is now safety profile. So the next thing I will do with these vaccine candidates is reversion to virulence. Also looking into safety profiles if you do dose effects, because some of the more promising candidates that had been put to publications, you never heard of them again, because they turned out to have a very small or narrow dose range where you could apply it. So this is one feature. If these vaccines candidates give us still good results in the, I would think that 5 to 10 years is a suitable amount of time. In my own experience for the CSF one that we licensed a couple of years ago, it took us 10 years from the promising candidate to licensing.
Okay. Well, thank you. And Elizabeth, I'm sorry, since my technical difficulties had me blanked out when you were talking and answering questions. I'll go to David first, and then I'll come back to you and ask you if I missed anything, okay? Because you can see the questions in the chat also. So David, you mentioned your lump skin disease virus vaccine created with a knockout of immunomodulatory genes. Can you say which genes were removed, and do you have any explanation for the reaction you saw with that virus?
Yes, thank you, Dr. Geary and Isabel Lewis, for asking that question. So yeah, it's obviously a very good question. So we were very undecided in the very beginning, which ones to target, because again, although there's quite a few genes and we know a number can be activated or even removed, initially you don't know what effects it's going to have on the overall growth of the virus. So what we did, we tried to remove some of the regions where looked like there were more than one of those genes present. And so we first targeted removal from, it was open reading frames 5 to 8, and I think it was 11 to 13. And we got varying results, and we found that the virus itself just did not grow nearly well enough to be considered then later on, if it showed good potential as a improved vaccine, to be then grown to the sort of level needed for commercial production. So what we then did, we had to narrow down our search, and this was again quite an issue. So eventually we targeted just a couple of those genes. So one was the interleukin 10-like gene that's present in the viral genome. And another one is the interferon gamma receptor-like gene. And some of the others we looked at were serpin gene and Kelch-like gene. So those are some of the ones we have been targeting. And I mentioned, of course, the superoxide dismutase gene, which has shown good potential. And the question then, of course, is, why didn't they or give the sort of desired phenotype as an attenuated virus that had good potential then to be taken forward? And I think I did sort of try to answer that to an extent, although we had a lot of discussions with our cellular immunologists and guys in this sort of area, even from the big poxvirus experts internationally. It's very difficult to predict, knocking out one or even two of these genes, what the actual phenotypic result will be. Simply because, of course, none of these genes just work in isolation. And you're dealing with the virus's own set of genes, and then, of course, the host genes as well. And we know the immune responses are very complex. And so, end of the day, the advice was, you've got to just try, take out one or two of these, and then just see what the end result is. And that's what we did. And so we didn't get the desired result. We used the field strain as the parental virus, but with the vaccine strain already existing, the one that we have here, the results were pretty good. And so we've got a lot of hope and taking that forward. And so that's pretty much where we are at the moment.
Okay. Well, how close do you think your vaccine is to actually being a marketable product?
Another good question. Well, as I again sort of concluded in the talk, although we've at proof of concept stage, some of the the initial trials, we really need to do quite a lot more for registration purposes, and that of course is your safety, your overdose, pregnant animals, this sort of thing. And so if we got the level of funding required, we're talking about, let's say, about three maybe 4 years as a rough estimate. So yeah, that's potentially where we are.
You think you're just going to run into the same issues that we talked with Sandra about GMO?
Yeah, so that's something we obviously have been considering right from the beginning, and we've even been engaging with a lot of the potential stakeholders in different countries in Africa where these vaccines have potential, like in Kenya, and the general consensus is a very good feeling that these would be very useful. And with engaging with the stakeholders early on, the feeling is that we could overcome definitely these sort of issues as a GMO, and it's pretty much it's a country by country basis. So you can't decide on a sort of blanket conclusion as to their acceptability. But the feeling was definitely that these countries would be open to vaccines of this nature. And of course, the good thing is these vaccines, again, vaccinia, canary pox and things, have gone before. And so it's not a completely new technology in that respect, just slight differences.
Well, so maybe this is a little bit of a segue for all three of you. And for, again, people in the audience, write in your feelings on these things also. So I'll put it out to all three of you. This issue of GMO is quite an impediment to the vaccine industry. And what strategies would you employ to possibly alleviate those, circumvent them, or whatever, to minimize that? As scientists, many times we don't do a very good job of really getting the word out to the public and making them understand, or helping them understand what these vaccines are and all. And maybe Hollywood's been bad for us too, and creating all scientists are madmen, right? So, and maybe we have to do a better job of making our positions known. But so, any of the three of you, tell me, what are your feelings on trying to alleviate this problem of GMOs?
Well, since I was already talking about it, if you don't mind if I just kick off, because
Go ahead.
it's what I sort of mentioned. We actually made sure when we were doing this actual work with the funding from the Canadian government that we had stakeholder engagement meetings where we invited from the public to all potential stakeholders in government, regulatory bodies, the scientists, to have open discussions on these sort of issues. And the results of that were generally very positive. And so any of these sort of questions and concerns were raised in an open platform. And so we took the stance of being open, engaging from the very beginning. And I think that's what we found to be very positive and helpful. Thanks.
No, I think it's absolutely what's necessary. Do you think this is all just people's not understanding the positions with GMOs, or do you think there's any politics involved? And if there is, we don't have to get into what politics are, but do you think it's somewhat political as well as irrational fears? I'll ask you that, David, and then I want the other two of you to chime in also.
Thanks. Well, my neck may be on the line here, but yeah, I don't think one can for a moment think there aren't politics involved. Yeah, and I'm going to stop there from fear of repercussions. But yeah, and I see what the others say, but I think always politics does play a role.
Yeah. What do you think, Sandra?
I think actually the COVID situation is helping us a bit because people got familiar with more of the vaccine approaches. We have so many vaccine experts around the world. But this was ironic, but in the end, we still have, I think it helps our business to a certain extent because at school and in open discussions, platforms have been discussed. And at least from my perspective, I can only say most of the farmers, they accept that for African swine fever, for example, there is no routine approach that works, so that we have to deal with genetically engineering or with genetic engineering. And I think the younger generation, at least in from my perspective, is more open. And what we really need is communication that it will not turn you into a pig if you have a GMO in the pig. And there are still some irrational things, and you will not solve these issues. But if you can inform the ones that are more open, at least for our business, it already helps us, I guess.
Okay. What do you think, Elizabeth? Do you have any other comments on this?
Yeah, I totally agree with what has been said, and I think from the COVID experience, the community is more aware of the benefits of some of the new technologies, and I think is helping our vaccine development field. But also, I think another component is the experience on speeding up some of the regulatory process, because for some of these novel vaccine development, the part of building up and proving in the lab is very relatively easy at this point to just move this platform forward. But the regulatory process are the one that are perhaps the one delaying the whole process. And again, I think that the lesson learned from COVID may perhaps translate into more streamlined processes for veterinarian vaccines for the near future. Some of the vaccine under development for foot and mouth disease relied on subunit and virus-like particles, and our safe vaccine, marker vaccine, that relied on modification of the genome or just portion of the FNDV genome being involved. And those, I think that most of these products are provided as a chemically inactivated vaccines, and therefore you are not injecting the product live, and that have some advantages. But again, I think this is a long way to educate the general population on these matters, and I think we are in better position probably than 2 years ago to move in that direction.
All right, thank you. So I know that the title of this is this whole symposium is Next Generation Vaccines, but I thought I'd just ask a general question. I know when we're all submitting grant proposals to agencies for funding, they always want to see something novel, what's new. What are your feelings about using some of the older tried and true technologies to advance vaccines, especially for high consequence pathogens at this point that won't hit regulatory impediments that some of these others will. Again, there was some talk today about subunit vaccines and just killed vaccines and all. So thoughts from anyone?
I would say that in the case of foot and mouth disease, there is room for these new technologies. And you just mentioned some of them that are actually under development in parallel to the vaccine platform that I just described. And they are at different levels of development. And they bring some of these gaps in the current vaccines being applied that have been recognized and it needs to improvement. And I think that there is future for those. And I think you have to introduce this concept of vaccination for a purpose. So we have different settings with different complexities and needs. It's not the same in endemic countries and those that are free of the disease, and the needs for the properties of the vaccine, not for, it's slightly not only for the vaccine itself, but for the control measures that are implemented. Those two need to be used in conjunction and be coordinated, and they must be different. So these new vaccines may have a room for fitting some of these purposes, whether you are trying to just control the disease or you are trying to eradicate the disease. Different features of this vaccine could have a benefit. And I think it's a very positive movement I could see from this development from 10 or 15 years ago to what we see as a more realistic approach to be used in the near future. That's my perspective from the FNDV field.
Okay, all right, thank you.
I can share. I mean, for ASF, it's a bit more complex, but at least also from the European perspective, there have been EU-funded research projects and also projects like IMISAPI that try to get these platform ideas marketable, so that you already involve the regulatory bodies, so that, for example, you have this platform licensing, and then you can put in the end, the needed antigens in. In the beginning, it was very slow because also the regulatory bodies, this was the first time they were approached with this kind of stuff. And it is difficult for them to get something like a workflow that has to be followed. But this road has now opened up, and I think this is quite important that we keep these the flow into this kind of platform approaches, that we have platforms that we know are safe, and that you can then insert also pathogens that are emerging, new pathogens, but also the old enemies. For ASF, we keep trying to also use the more modern things, but at the moment, back to the roots work better than applying the most recent developments. In the end, the GMOs, these deletion mutants, are somewhere in between, and I hope that we can somehow link and combine these approaches, at least in near future.
Okay, thank you. Dave, do you have something you want to add to that?
Yeah, thanks. I think Elizabeth and Sandra summarized it very well. And I think every disease is unique in its own aspects and respects, and every situation or country has its own unique set of regulations and requirements. Again, the whole aspect of endemicity or not. And we can see that very much for lumpy skin disease as well. I mentioned a couple of the issues in non-endemic countries. First-time exposure, even with some of the vaccines, and there's so many available. So definitely, use of a subunit, use of inactivated, they have their places as well. But as I try to also indicate for the African situation, there's definitely room and scope for something like the vaccine technology and this multi-pathogen approach in a single vaccine has a definite place. And so I think every situation is pretty unique, and you have to evaluate them on that and just see what can work and what can't.
So speaking of all the different African nations, there must be a lot of different feelings and regulations for many of the different nations in Africa, isn't that correct?
Well, yeah, there are, but if you know the AU-IBAR as the big sort of overriding body for much of Africa, for a lot of these concerns with different vaccines, diseases, and so there are the bodies that actually control, so we say, or regulate to an extent in different countries. And we have SADC here, Southern Africa, you have the East Africa side. So there are these organizations existing. Of course, then we have the FAO and the OIE to really help with these sort of processes to get acceptability and uptake in different African countries.
Okay, thank you. Well, I guess we'd be remiss if we didn't hit on this topic now. We've all seen the tremendous speed of development and high degree of efficacy demonstrated by the new mRNA vaccines. It would appear that the animal health world is not ready for these vaccines yet due to logistics, refrigerations, administrations, whatever else. Do you see a day when some of this technology may be applicable to animal health vaccines? And if so, to which ones? Who wants to field that first? You're on screen, David. You want to?
Oh, no, ladies first, please.
Actually, I don't know. I think Dr. Rieder already answered it a bit. So for the RNA viruses, I see application, and in the end, it feeds into our toolbox of possible vaccine approaches. And for these vaccines, I really think that this is feasible. As I said, unfortunately, for the large DNA vaccines or the DNA viruses, I see that there is more room for at least decent research before we can use these approaches. I'm not sure if you are of the same along the same lines, but I think for the RNA pathogens or the viruses, I see immediate application. For the large complex DNA viruses, we need more knowledge about the antigens, I guess.
What do you think about the logistical issues?
In the end, if you look into if you use a live vaccine, this is also you also have to manage it. This is one of the reasons why it fails in some countries. And for Central Europe, I think if you really want to use it, you can do almost the same effort that you now did for human beings. But in remoter areas, this is definitely an issue.
Right. Elizabeth, when I was in the technological black hole, did you discuss this already?
No, what is your question?
About the possibility of using the mRNA technologies for animal health in the future?
Well, I could just comment that I'm only aware of some recent efforts on that. There is no publication, at least for FNDB, or I haven't seen any presentation yet, but indirectly at one of the recent EOFMB, there was an indirect comments that efforts on that directions are being pursued. Of course, the cost of goods would be one item to consider compared to other platforms being under development, and also how that address the diversity of strains. As you are well aware, when just the cross protection, even within a serotype, is very limited. So making one strain not necessarily would protect against others. So what is the target of expression? How the diversity of virus like foot and mouth disease is addressed by those platforms, there is still a lot to do from the research point of view. So not having any evidence of successful with our system, I cannot predict, but I think it's definitely an interesting area of research to pursue.
Thank you. David, do you want the last word on this?
Thank you. I feel a bit guilty here, but yeah, I think again, Sandra and Elizabeth have said it very well. Yeah, and just bringing a point, a few years ago, we were doing a bit of an analysis to see what is feasible for, like, in Africa for any of these new vaccines. And the figure came up that feasibility-wise, for your average, let's say, emerging farmer, a vaccine cannot be higher than about a cost of $1 a dose, and that just would not be practical. That was the figure that was used. If it was a few years ago, it probably hasn't changed very much. And so really affordability is a very big factor. And that's why, again, we think the sort of platform technology we've been working on has a lot of potential. Again, the vector being very stable, and of course the whole issue of your cold chain maintenance, it obviously comes in there as well. And so having a stable platform, low cost, already been in production and well characterized, that can overcome a lot of these issues, is something obviously very attractive. Thanks.
Yeah. Well, as you mentioned, cost is a big factor when you talk about animal health vaccines, but some of these very devastating pathogens, all that we're talking about today, maybe people and governments are willing to pay more. And I'm out of my element on this aspect, so I'm going to ask the question. Sandra, with ASF, due to the terrible economic consequence that it has on countries, do you know anything about like the pricing? Or would governments be more willing to pick up the cost of these? Or yeah, maybe I'll end it there. Why don't you tell me what you think?
It's a difficult question. I can only tell that in the end, for the German situation, we have these kind of insurance systems that could also cover vaccination. We so the farmers pay into partly state not funded but state governed insurances. And if you have an animal disease outbreak, you could either get refunds for culling, but they could also cover vaccination costs. And under these circumstances, where you have a compensation scheme in place that could also deal with that, pricing is not so difficult. So it could be much more expensive if for the sake of a whole population. But still, on a global or even a European scale, I think pricing is something that is important. It's a notifiable disease, ASF, and it is, in the end, a control program that will come through the federal governments. So I think it's a political decision if you want to go for vaccination. For CSF, we know we have vaccine banks. If we have something like that for ASF vaccine, I could envision an approach where moderate pricing is still acceptable. It's not about $1. It's probably whatever, 5, 6, 7.
Okay, all right. Do you have any comments on this, Elizabeth?
Yeah, in the case of FNDV, this next generation are coming with an advantage that could potentially reduce the cost of food. For instance, our FNDV leaderless marker vaccine comes with the DIVA marker that is embedded or encoded in the genome. So you could actually process just a crude preparation without the needs of downstream processes that are normally used on commercial vaccines to eliminate the non-structured protein and purify the antigen, the capsid antigen. for the vaccine production that is DIVA compatible. The fact that we have this DIVA marker encoded, you could just produce a relatively crude preparation, inactivated, and have a good product that potentially could reduce the cost of production. The fact that we work with the attenuated and safe vaccine candidate has also allowed to downgrade the level of high security levels, BSL-3, and some other platform experience that as well. For instance, BLP or other ADNO FNDV platforms have similar situation where the production can be done at a BSL-2 Ag plus laboratory without the needs of BSL-3 regulations. So that's what I could respond for the FNDV.
Okay. Well, one of the people from the participant pool has said that the biggest gain from the COVID pandemic really is the success story of using RNA vaccines for the first time, and how this opens up many opportunities going forward for all sorts of therapeutics and vaccines. Did make the disclaimer, he doesn't work for Moderna, so that's good. So I think there is a lot of, there's a bright future. But with that, I'll ask you a broader question too. What may be next? Have you envisioned or heard of, or any other platforms or approaches that may be applicable in the future? I know a lot of our gains in the animal health world tend to follow what's going on in the human health world, but any speculations as to what may be coming down the pike? You can say no if you don't. I'm going to take it from the silence that it's a no. So
it's
we'll
rather. I think what we have at the horizon is talking about other delivery systems. We know that in the end, this kind of intradermal and all these kind of approaches are now applied, and they seem to hold promise also for the veterinary world. But I'm not so much into detail that I could give more than just the wake. Ideas.
Okay.
I just want to. Can I just comment on
sure.
this question? Of course, I want to see our platform moving forward and being able to do just more testings with master and pre-master seed virus vaccines available when the scaling up and production take off. But I think moving forward to next development would be important to see if the combination of vaccine viruses with different adjuvants or different biotherapeutic molecules could improve the duration of immunity for some of these vaccines, especially for FNDV. We know that the current vaccines of good quality, whether they are oil-based or aqueous-based, they have a limited protection level up to 4 to 6 months, and then you need to revaccinate. So having a product that extends that time to longer times could be a desirable development. We know that the outcome of infection induce a much stronger immune responses to the virus, and that is long lasting. So it has been an effort for many years to try to mimic that by doing formulation that could enhance those properties. So that is an active area of research in our laboratory and around many other FNDV laboratories. But I think the key would be to have the basic knowledge and understanding of what are the determinants of protection and duration of that immunity. And when we identify those, we could help up by incorporating in the formulation as an adjuvant or or a molecule in the formulation that would allow us to extend that level of protection to longer period of time. Of course, under experimental design for foot and mouth disease, they're always in the back of our mind and efforts toward looking at live attenuated vaccines, but just to understand from the basic point of view, what are the bases of protection? You know, it's still a long, long way to really have any promising development on that, or perhaps not. But I think those type of experimental vaccine and knowledge from the research groups would be very fundamental to bring this improvement into the pipeline. So that's an important area. The other, at least for foot and mouth disease, you have a window where the onset of protection given by a vaccines is not provided, and that comes from early times following infection. Following vaccination. So there have been efforts also to look into biotherapeutic to just fulfill this need to bring protection on those.
Okay, so I thought that was kind of, I think she was saying also, I believe, was that an issue that's very important is to develop immunity as fast as possible, especially in these emergency use vaccines, such as FMDV would be here in the US, and I assume ASF also. Methodologies and formulations that will induce protective immunity from vaccination as rapidly as possible is an extremely important component. I believe that's what you were referring to in the end. Is that not right, Elizabeth?
Yes, sorry for the analysis background. Yes, definitely interference or other biotherapeutics that could, you know, fulfill that window of lack of protection would be important, especially, you know, in disease-free countries where they would like to, you know, improve the coverage given by the vaccine. So I hope that answered.
No, that does. And I think, David, weren't you trying to say something there too?
Thank you. Once again, Elizabeth and Sandra have done a brilliant job at covering most of the bases. And yeah, I think from our side, again, the whole thing about the adjuvants and, you know, just getting a handle on the immune responses to these various diseases and the vaccine technologies we're using. And again, the field of nanoparticle technology and slow release sort of vaccines, those are, you know, quite a very relevant and interesting area that we so you could almost get a prime booster effect in one inoculation. And so that's something that's something would be really advantageous. And then, of course, the whole thing, especially like being in the cancer field, is then sort of specific cell-targeted approaches to be able to deliver these vaccines to specific cells. And again, with the whole thing of the inflammatory responses and things, to try and modulate those to the extent that we don't get some of the rampant responses that we have seen. And so, you know, getting that under control is really important and an exciting field. Thanks.
Yeah, thank you. Well, since the three of you have listened to each other's presentations today also, did you have any questions for each other that you want to ask? David, if you don't want to answer, you better get your face off the screen now, all right?
Okay, well, I've unmuted. Yeah, the one I think Sandra knows quite well what I'm talking about. At our IABIS technical committee meeting not long ago, there was something about for ASF vaccine, I think from Vietnam, if I'm not mistaken. Sandra, I think you said the upcoming webinar on that, is that where that's going to be discussed? Because that sounded quite exciting. Again, that's not my field, but it's something that I picked up on. Of course, at Honest to Put, well, ASF research by other colleagues is certainly very important. I don't know if you want to comment on that or not, but that's just something that piqued my interest.
I have actually already covered part of it. In the end, the Vietnam issue is the USDA vaccine candidate I-177L. This is the candidate that was or has shown really high potential in experimental studies, and Vietnam has started field trials where it is still very promising, but a bit less sterile immunity than under experimental conditions. But we invited all these, or the body, to join our webinar that I put on my last slide. So there will be a GARA IABS Star IDAS webinar on ASF vaccines on the 6th of May. Actually, so I really re-invite everybody, because what we want to achieve is that we open the discussion on what do we really want to know from the vaccines, what is the acceptability. So is there side effects that we can tolerate if they have outstanding efficacy, for example, or for certain settings? And in the end, Haley put it in the chat right now. So there is a scientific part. So we will give an overview on the most recent viruses that have been published, including the one from China. And then there will be an open discussion with regulatory bodies regarding the way forward.
Thanks. I do apologize. I missed that part of your talk. There was an interruption that I had to deal with. So
Don't
my
worry,
apologies.
it's always the case.
Thanks.
So as I said, with all these live attenuated vaccines, we have to talk about what can we accept? What's the risk level that we can accept for a vaccine strategy?
Thank you.
So also a question for Sandra. All these live attenuated vaccines mark, that they could be traced by DIVA test, and are those already developed for companion tests for these vaccines?
Some of them would have DIVA potential. Unfortunately, the most promising ones are not the DIVA compatible ones, at least at the moment. They, of course, at the moment, they still have marker genes inserted, so that you could, at least in the field, you could find them just by PCR screening. But from a serological point of view, there's room for improvement. The problem, or the thing was that we never really had true candidates, and DIVA was not the biggest problem. So the problem was to achieve protection at all, and also safety. At the moment, this is the next step. But in the end, at least from the genetic point of view, you could distinguish.
And another question is regarding the onset of immunity conferred by these vaccines. Are all sort of very rapid? I always get very impressed by how rapidly these vaccines could elicit a protected immunity in pigs. So are is that a very important feature that everybody look into the vaccine, or is not as relevant as having a full protection and other features you expect in a prototype?
I think it's depending on the situation. For the emergency vaccination, we would really like to have early onset of immunity. The ones that I tested have protective effects from roughly 7 days post the first vaccination. But this does not give sterile immunity or something. It really protects the animal. And if you look into the R note or something, they will reduce transmission, but they will not stop it at the first go. I think for the situation, endemic situation, for me, the safety features are more important than the onset of immunity. But to be honest, I think that ASF has a problem with duration of immunity. There we already have some indications. The onset will be roughly 7 to 10 days, I guess.
Well, I think this has been a great session, a lot of good and open discussion. And I want to thank everyone for participating in today's symposium. And I'd like to also encourage those of you in the audience to consider becoming members of the U.S. Animal Vaccine Research Coordination Network, so that conversations such as we've had here today can continue, and your opinions can also be heard on other issues of importance to the animal vaccine communities. The link to this network can be found on my biographical sketch, which is included in the program. And Haley, you're going to put this out to everybody also. Can you put that in the chat now so that I see you did. There you go. Thank you very much. So I would encourage you to go to that website and become members to continue to have just such lively conversations and important conversations. So when you log out of today's session, you'll be prompted to complete a post-webinar survey. So please take a moment to fill this out. And finally, I hope you'll rejoin us tomorrow for the session on epidemiology and disease ecology. I want to thank everyone again and have a nice remainder for your day. Goodbye.
Thank you.
Thank you.
Thank you.